So welcome to my presentation where I'll be talking about the comparison and benchmarking of combinatorial chemical spaces and enumerated compound libraries. I will start with a very bold claim. I have to organize a little bit because I don't see anything. It's not much better. My claim, or the claim of our company, is and was and will be chemical spaces are better. And to be fair, this is not something that was proven before. It was just something that we perceived internally, also that we perceived when working or speaking to customers, that chemical spaces come with a lot of benefits. Unfortunately, there was just nothing that really was kind of like written down and peer-reviewed and really telling, hey, chemical spaces are the better choice. But there were kind of like hints around. So right now, I think this is the consensus we have to agree upon. Bigger is better. So the larger your compound collection is, the better the results will be that you can retrieve from the larger collection, which kind of like makes sense. Assume you have all the molecules available and you're looking for the perfect molecule. So obviously, if you have access to every single option, you will be able to find the best molecule if your algorithm allows to do so, right? And the problem is though, so now we have the chemical spaces and we have the libraries. The chemical spaces are larger and the consensus is larger the better, but we still have the libraries that have been on the market for decades, right? And the question is, well, who is the David and who is the Goliath in the whole scenario? And I think the answer is not that straightforward or not that simple. Ultimately, since libraries have been on the market for decades, they come with very well-known formats like SDF or SMARTS, and they have been the standard for decades. So every industry just uses SMARTS and SDF. They are quite universal. So basically, any software that works with ligands somehow can process either SDF or SMARTS or both or any other known formats. And what the beauty of those formats is, well, they are very straightforward. They're simple. Basically, you open up the file, you see the SMARTS, and you know which molecule is inside this file, right? So we come up to a point where we have this very reliable solution that has been used by generations of computational chemists since, I don't know, the 80s, 90s even. So there is no risk by using and continue using them because every software is able to utilize them, and therefore everything is a little bit, rigid. It doesn't. This whole system relies on those old formats of these libraries, and therefore it's very hard to adapt to new formats. But if you want to progress, if you want to take it to the next level, you need to do something else, and this requires a bit of courage, So this is where a colleague of mine, Raphael Klein, and I sat down and discussed things and actually came up with the idea, well, let's do it. Let's sit down, let's discuss the whole topic, and let's find out what is actually better, chemical spaces or libraries. And we did it successfully. It was published in August. And this is the publication, a benchmark set of bioactive compounds for diversity analysis of compound libraries and combinatorial chemical spaces. And this talk is actually just what it is, the publication, and I would like to guide you through our thought process and also to guide you through the results to make your own conclusions. So the whole idea was, okay, let's compare libraries and chemical spaces. So we have apples on the one side and oranges on the other side. The main problem is, okay, how do we compare them? But ultimately, you need compounds to do so. The most obvious choice, and this is what you can see in most CompChem papers, is basically, just use the FDA-approved drug list. I mean, it's applied in quite a few publications. It's pretty straightforward. Those are drugs, so therefore, why not? Well, the caveat with FDA-approved drugs is this is the creme de la creme, right? Those are the best compounds that survived several clinical trials. They survived a myriad of optimization steps. This is something else that the normal compound you're working with, right? So what is the most important aspect of those structures is they are extremely complicated, right? You had to optimize every single heavy atom at one site, at one point. Everything is carefully selected. and therefore the synthesis steps are huge. Usually it takes 6 to 7 synthesis steps to come up with your final compound. It may be reduced once this whole compound really enters the approved status where scaling up is a thing. But nevertheless, it's quite rare to reach an FDA-approved drug with 1 or 2 steps. And this is kind of like the trigger point that we wanted to achieve. It's quite easy to add FDA-approved drugs into library. You open up your file, you add a new FDA-approved drug, and there it is. It's already in the library. Chemical spaces, as I've mentioned, work differently. You cannot just simply add molecules. You have to add the building blocks and reactions. But as I may reveal to some of you who don't know yet, chemical spaces are a little bit special. They usually employ 1 or 2 step synthesis steps, which limits obviously the complexity of the final compounds you may retrieve. So it's not possible to just use a drug that has 6 or 7 or 8 synthesis steps and cover this whole thing with 2 steps, right? So we wanted to have a set that really reflects the chemical landscape a little bit better than FDA-approved drugs do. So this was where we sat down and we decided, okay, let's use the Campbell, right? Campbell is publicly accessible. We can argue about the quality of data, but nevertheless, it's still in the higher tiers. So we took the Campbell data with approximately 11 million entries. And those entries contained potency, IC50 values, you see everything in pinkish on the left side. And we did some systemic filtering regarding, okay, what would you like to have by the end of the day? So what we wanted to have is a set of relevant bioactive compounds. Relevant means for us, it's not something that is in the micromolar range, double-digit micromolar range. We wanted to have the good stuff. And the good stuff is something with biological activity less than 1000 nanomole. Also, we didn't want to limit ourselves too much on the drug likeliness. So we extended the molecular weight up to 800 gram per mole. We didn't want to have too small structures, so everything with a size less than 10 heavy atoms was filtered out. Since this was more in the direction of working with small molecules, we also excluded microcycles. We also excluded off-target activity. This is something I see quite rarely when people are actually working with the Campbell dataset, where they just screen for IC50 values and then proceed. The problem is, though, you also have off-target effects like CYP enzyme inhibition and so on. So this is not data that is bioactively relevant for you, right? The subsequent steps, basically anything from 6 to 10, was a cleanup of data. Sometimes entries were just kind of like more than or far less than 1 nanomole or something, which is not extremely precise. Duplicates were removed and so on and so forth. And after we did the whole systemic filtering and processing, we ended up with set L of our benchmark cells. So we actually decided to have 3 different sizes, each that can be used in different areas of computational chemistry based on how many computational power you have on hand. So you can see with each step, it shrinks one potency less. So basically 10 to the power of 5 for set L, 10 to the power of 4 for set M, and finally thousands for set S. So we came up with a set L, approximately 380,000, yes, correct, of compounds. And this was everything that was biologically active. So the problem is, though, now we have a set also with a lot of compounds, right? So sometimes you end up with a lot of scaffolds, similar scaffolds, same scaffolds, because all of them were quite potent. And that's why we decided, okay, let's just work with one compound class, because this is not diversity. If you have, I don't know, 5,000 molecules with the same scaffold, this is not a nice set to work with. So again, we went into the Birmes macroscaffold clustering and just picked the smallest representative of each scaffold set. This let us reduce the compounds quite nicely to a size of 25,000 of set M, M for medium. And then we went further. This is where I would like to go a little bit into detail. So in the shrinking of set M to set S, we wanted to have the most diverse representation of the chemical landscape of bioactive compounds. So what we did here, first thing we did was a principal component analysis. So ultimately taking a lot of molecular features into account, we wanted to reduce it into a two-dimensional world. So basically we then calculated PC1 and PC2. And what you can see here is that we ended up with quite a spreading, right? This is not something so nice to look at. So what we also saw is that a lot of compounds have very exotic structures, functional groups. And this is also something that we didn't want to have, because this is not something that really reflects, a bioactive drug-like compound. So what we did, we actually trimmed everything. So anything that we decide is an um, exotic compound, meaning exotic means in this case that it contained very extreme structures or extreme physicochemical properties, as you will see in the next slide. So we took the top and bottom 2.5% of each principal component and cut them off, ended up with a zone of set S. So basically, if you now take a look into the exotic compounds numbered from 1 to 8, yes, you will see what I mean. We have very linear compounds, not very drug-like. We have very flat compounds, like compound 3. I will also use the pointer here. So this is very flat. This is not something you would consider drug-like at all. We have very sugar-like-ish compounds. So this is where you would say, well, this is not something that you would do in a lab synthesis on a daily basis. So this is something where you would actually consider some biomechanical processing, bioengineering to do the bacteria or the yeast for you. And compounds like this, where I have to ask myself why it's bioactive at all. And you also obviously end up with a lot of peptidomimetic compounds. This is a real peptide here, or even compounds with a nucleotide structure like this compound here, where we can also again disagree if this is a drug-like compound with that many negative charges. So ultimately, looking at those extremes to reflect the idea of removing exotic compounds, because this is something that should be included in a library per se, we proceeded further. And then we did a segmentation. So basically, we divided zone S into a 10 or 10 times 10 matrix. And from each zone, we extracted 30 compounds. The idea here was to actually cover a very diverse set of compounds that is also homogeneously distributed in the zone S based on the principal components, which lead us to this beautiful visualization, which also can help us to understand the chemical landscape of the benchmark set. What we did next, also, which obviously took a look into each of those zones, and we call them quadrants from 1 to 4. And each quadrant was very, so the limits were obviously fluent, but nevertheless, there were some characteristics of each quadrant that were quite prominent. For quadrant 1, it was obvious that we had very small and less complex compounds with drug-like character that can be seen on the left side. On the right side, we had an increased hydrophilicity with the highest average of H-bond donors and acceptors, and closest to the lead and drug-like molecules. And now here this is an example for compounds of this section. Q3 was the most lipophilic one. It contained a lot of directly connected aromatic ring systems, which you can see here. So you can also imagine the flatliness of compounds in this area. And Q4 contained the most complex compounds with the highest frequency of beyond rule of 5 compounds. So again, kind of like handy to really get an idea for the mapping of the benchmark set. And if we separate a little bit further, we can also see that this region covers fragment-like structure, mini fragments, nucleobases. In this area, we find a lot of aminoglycosides, peptides, and nucleotide structures, not as extreme as the one example I showed you before. We have a very lipophilic area, which contains a lot of carbon-rich ring systems, where nitrogen is a little bit rarer to say, far less H-bond donors in this area. And here it becomes a little bit wider, so we have a lot of natural-like compounds like opioids or steroids. and a lot of sp3-rich compounds. So this is also the area we saw the highest number of beyond rule of 5 compounds. And the sweet spot, so the area that we are used to, is actually in this area. So this is where we can find a lot of the drug-like compounds. So maybe a little bit of insights how this area came to be. So we obviously checked also the distribution or the physicochemical properties of the distribution here. And if you take a look into the molecular weight, you can see from the light pinkish color here, which is always the lowest, to the dark green one, which is always the highest for each of those parameters. We can see a nice gradient following here. So basically small compounds and large compounds. Same goes also for log P. We have the least lipophilic compounds in this area and increasing lipophilicity towards this area. As for the numbers of H-bond acceptors, least amount of H-bond acceptors in this area, in this section, with an increasing character towards this direction. And the same goes for H-bond donors. Almost no H-bond donors and increasing number of H-bond donors. And every time we also had the beyond rule of 5, the Lipinski's rule of 5 parameters. I drew a line for the cutoff. So basically anything on the right side here is outside of Lipinski's love. So basically disobeying Lipinski's rule of 5. Same goes here. This is the bad area, bad area, bad area. And if we now overlay all of them together, we actually can see that anything that is not touching those areas, basically this section, is what we can accept as a drug-like section of our benchmark set. Okay, so we now have the benchmark set, so now we can work with that quite nicely. We also took a look into the distribution of targets, and it's actually quite similar to FDA-approved drugs. So therefore, we did a lot of assessments here that I want to go into detail, and ultimately, just to the take-home message here is that the benchmark set was well suited to be employed on different compound catalogs. In the next step, we decided, okay, let's compare now our chemical spaces, and we worked with the chemical spaces of our partners, basically N-Benta, Ottawa, E-Molecules, ChemSpace, Wuxi, and Enamine, and also here the overview on the sizes back then. So this is like 1 year ago, I would say. And we need comparison, right? So we took a look into other catalogs that are not covered by them because it doesn't make sense. Chemical spaces usually are the ultimate potential of a compound vendor, so all the possibilities of compounds that can be synthesized, and enumerated sets are usually just a subset of the space itself. So therefore, it didn't make too much sense from our perspective to take Enamine's real database because it would be just a waste of time, honestly, because Enamine's real database is just a subset of the real space, and therefore comparing apples and oranges didn't make too much sense here. So we decided to go with other compound vendors just to get a feeling for this size, actually, if the size plays a role of the quality, of the results quality. So we worked with ChemDiff, Life Chemicals, Smallpot, and MCure, and downloaded the libraries. And that can be seen here. The smallest one is 1.5 to the power of 10 to the power of 5, which is 150,000, and then increasing to almost 6 million with MCure. 6,150,000 compared to billions and trillions in this section of the chemical spaces. So almost 1,000 times larger. From the largest library is 1,000 times smaller than the smallest chemical space here. This is where I love to be quite transparent. Caveat here is obviously we are working with just selections of compound libraries. We always tried to go with the largest one that we were able to download. And here I have to mention it may look different for other providers or other subsets or of the libraries that we investigated. Especially here, I have to admit, we're working with a representative diversity library. So ultimately, if it's a diversity library, I assume that it will also reduce to contain only a quite diverse selection of compounds, and therefore, if this library would not be diverse but cover all compounds that ChemDiff has to offer, also the observations would change in the long run. So take everything with a grain of salt, but nevertheless, I think you will get a feeling for the development and the trends here in the upcoming slides. So what we did, we took the set S, the small one, and employed all three of our algorithms on all chemical spaces and all compound libraries, and retrieved 100 results with each method for all of the 2,917 compounds as queries here. So basically, to translate it for everybody who's not familiar with our solutions, for each of those queries of the 2,917, we retrieved the most 100 similar compounds based on each algorithm that we have developed. Here you can see the scores for the top results, so rank 1, the top result, and at the bottom we see the results for the top 100 results, so all of them and the best one. And what you can see for the top results, in almost all cases, chemical spaces were performed better than the investigated compound libraries. MQ, though, was also able to compete with the chemical spaces. The trends remain more or less very similar for Spacelight. For Spacelight, we employed two different fingerprints, the most well-known ECSFB4 and one fingerprint that was developed exclusively, not exclusively, but especially to screen on our combinatorial chemical spaces, namely FCSFB4. And the trends among all three methods remained ultimately the same. So chemical spaces were better than compound libraries, but MQ was able to keep up with the chemical spaces, at least for the top ranked result here. This changed a little bit when we look into the mean of the top 100 results. So basically, we took each score of the 100 results, calculated the mean, and then also put it on average for every single query. And what we can see here obviously decreases, right? The top rank result is obviously better than the mean one. And what happens here is, while the decrease is quite low compared to the top rank result for the chemical spaces, you can actually see how the scores for the chemical spaces drop significantly. And this is, I mean, take a look at ECSFP4. This is the most well-known one. As we can see, while Explore and RealSpace are able to keep up with a pretty decent ECSFP value score in the top 100 results, we can see that all libraries drop significantly, and especially the smaller libraries, we already reach scores where you can question yourself if this is still a similar compound to your query. A very interesting aspect here, we also have those error bars, which basically reflect the standard deviation. And those are very nice indicators for diversity. And if you would look into the numbers, they're also published in the supplementary information of the publication itself, you will notice that the standard deviation for the chemical spaces is significantly lower compared to the libraries. This means that the results that you retrieve in those 100 results are themselves similar to each other, or at least the score doesn't drop that much, which is an indicator that the results that you retrieve for each method are in itself not so badly distributed. So ultimately, here you end up with falling way faster. The score drops way faster for libraries, while for the chemical spaces, the drop is way, way smaller, which means you get better results. But you will see that in a few slides as well. So if you take a look at the F-tree similarity, it's just some random pics. You will see the whole pictures in the publication. It's open access, by the way. What you will notice is that the distribution of 1 is the highest again, and 0.9 is the lowest for the coloring. You'll actually see that for F-trees, for the benchmark set, and this is now the mean similarity of the top 100 results, we actually see that we are in this area ultimately. So it's pretty rare for us to drop. We have the best results in this area, which is the rule of 5 area. So for the drug-like compounds, we actually managed to cover it quite nicely. Explore seems to be has the best coverage. Next would be real space. But in the freedom space, we already see that, especially in those areas of the beyond rule of 5 of compounds or the more complex compounds, we see that the score starts, that the coverage becomes far less dense. Yeah. It becomes more evident if you now compare Explore to MQ and MPort for the top 100 results. We can see that the coverage almost disappears. So there's a huge emphasis on drug-like structures, I would say, in this area, whereas for everything else, the coverage is almost gone. Again, I'm a transparent person, and the caveat here, obviously, if you design a compound for drug-like compounds or a library, you end up with excluding two undrug-like compounds. So basically, even if you put a cut of a 500 Dalton or 600 Dalton, you basically eradicate all possibilities to cover those areas. So it's not like it is bad, bad is the wrong word. It's not like it is designed to be this uncovering, but it can be a consequence of the design of the library itself. This is now the ECCP4. We see coverage becomes a little bit lower. The lowest is 0.4 in light. You can still see somewhat occasionally points, but it's not as high as we expected for FTs, but also the numbers for each score are different. So for ECCP4, we decided that 0.45 is a decent cutoff. F3s has just way higher scores in general, right? The scores of F3s are more closer to 1 than they are to 0, whereas for issues of P, this works a little bit differently. So ultimately, just the coverage, we can see it's occasionally broader, and it becomes more evident if you now compare them with the chemical space, with the libraries. We can see that the coverage again drops significantly for the libraries. Spacemax, same observation ultimately. We see a nice coverage for the chemical spaces. And if we compare them to the libraries, again, the coverage differences become apparent. How does it look like in a better distribution? So ultimately, each of those, this is the violin plot for each chemical space and each library. We can see that each plot is also colored in the mean value for ECSFP4, and this is the distribution of results. So ultimately, what we will see for Explore, we see that towards the better ECSFP4 values, we have a broader distribution, so more compounds are in those areas where it become more relevant, more similar to our query compound. Same also for real space, whereas for the chemical libraries, we see that obviously the heavy point is a little bit to the left distributed, and it's also quite narrow in the better sections. Same behavior ultimately also applies for Spacemax, so there were no surprises for us here as well. What's interesting though is the development of the score ranking. So here we can see now the ECSFP4 score, the molecular fingerprint. The first data point is actually the mean similarity of the rank 1 results. And you can see that all chemical spaces start quite highly. And what we can see here, for all of the 100 results, we formed clusters of 20 compounds and calculated the mean score for each cluster among all results. And obviously, the score drops. I mean, this is the nature of scores. You always have the most identical or more similar compound, and then all the least or less similar compounds come. And you can see here quite nicely how the differences between the chemical spaces and the libraries are. You can see the steep is lower, so it's not dropping that fast in the chemical spaces as it would do in the molecule libraries. We already see here, we end up with ambrosia as the worst space in this section, where we already dropped below 0.4 of the ECZP4 score in the least interesting cluster. But in the case of the chemical libraries, all of them end up in this section, right? So ultimately, if your cutoff is at 0.5, you can see here, if you still manage to deliver you at least 20 compounds that are relevant, where in the case of chemical spaces, it's no problem for all of the spaces. Same observation can also be made for spacemax. The drop is stronger for the libraries as well. Okay, so this was actually a nice anecdote. So ultimately, this is what we did after the first reviewer complained. We decided, okay, let's do an additional assessment to really put it into numbers, to really have a feeling for what's the capacity of a chemical space to provide you with relevant compounds. And what you can see here are only the libraries. What we did here, we took all results of the ECSFP4 screening runs and calculated how many of them are able to, how many with a score higher than 0.45 are available. So how do you read this? Ultimately, for ChemDiff, only 4 out of the 2,917 queries provided you with 100%, so 100 results, where all of the results had an ECZP4 higher than 0.45. Again, in only 4 cases for those queries, only 4 provided you with 100 queries, 100 results that had a score higher than 0.45. If you go to how many of them actually had 50 or more results with a score higher than 0.45, it was 10 for ChemDiff. And those numbers continue for live chemicals, Molport, and also MQL. And you can see MQL is able to outperform all other spaces, and it was also the largest one we've been working with. Then, now the chemical spaces, we see that ChemWire is able to outperform all other 3 libraries in this scenario, but performed worse than MQL in this comparison. Next are Galaxy and Ambrosia, almost similar behavior, I would say. At least 440, 450 queries ended up with 100 results with a higher score. So basically, this is an indicator for what's the power of providing you with more similar compounds. And this is where the chemical spaces now start to really set off. They outperform you with the ability to provide you with more relevant structures. Next, we had Freedom Space and the New Simple Space. So this is not published. This is the first time I'm showing this to anybody. Simple is a little bit outperforms Ambrosia and Galaxy, but is a little bit behind of the Freedom Space. And everybody who did the math, you know that Explore and Real Space are the only ones left. And now to say who performs better, it is Explore. One can argue what the difference between both are, but at least in our investigation in this scenario, with those versions of those spaces, Explore was able to outperform every other chemical space. And in general, we also have to agree that chemical spaces outperformed libraries again. So to put those into numbers, basically all of those numbers shown here in this, let's phrase it chaotic representation. If we now reduce it to just bar diagram, we can see here that the chemical spaces are able to provide you with more compounds, more similar compounds than the investigated libraries in this case. I'm sorry? Oh, good now. Takeaway for you here is ultimately, we just looked at 100 results. Yet there will be more, thousands, maybe even millions of compounds that are more similar. So if you're working with chemical spaces, you are able to retrieve way more structures that are similar to your compound compared to the chemical libraries you have here. If you have something that really starts to match your compound, you will have a ton of access to more compounds here. So ultimately, the takeaway is if something is similar inside the chemical space to your compound of interest, there will be a ton, not even a ton, maybe more millions of compounds that are similar to your compound of interest. And if you think about it, it's not about finding the best compound, the exact same structure, right? It's about finding something around the compound of interest. We also took a look into the quadrants as we have described, and also took a look into the rule of 5 compliant compounds. And ultimately, what you can see here for rule of 5 compliant compounds, the takeaway is that chemical spaces provide you with better average scores for rule of 5 compliant compounds than the chemical libraries do. On the landscape coverage, this is a density plot. We will see that Explore is actually nicely covering all 4 quadrants, and almost every single compound collection we investigated had a heavy emphasis on this area of the rule of 5 area, whereas real space seems to be more trimmed, so it's more focused on the drug-like compounds, which you can see since there's almost no coverage for those zones. Freedom space, again, the behavior, though, takeaway, Explore does a better job in covering all 4 quadrants. If you compare it to the chemical libraries, surprisingly, MQL is also able to cover those areas as well, whereas Molport, for an example, is quite focused on the rule of 5 compliant compounds. Next to the clustering, because time is running out, just to give you an idea, we obviously investigated how the search methods perform. So if you have a search compound and we are using our fingerprints, you can see that the fingerprints quite nicely assembled around the compound of interest. Also for Spacemax, Spacemax is also structure focused. So basically the sequence of heavy atoms plays a role for Spacemax, and therefore those methods are actually able to find something in the proximity of a compound. But as we have heard, F-trees is quite orthogonal. So based on U-mapping, again, similar methods to principal component analysis, where you put several dimensions into one plane. We can see that F-trees is quite orthogonal, so it's far off. The results are not similar from a finger point of view. So they form their own clusters. And as we have heard yesterday, actually F-trees is quite able to provide you with high quality chemistry. So I am not a huge fan of fingerprints. It's like the best thing we can, it's the most common thing that people apply, but F-trees is quite orthogonal to this method and therefore provides you with something a little bit outside the zone, but that is still chemically related to your structure of interest. And if we now compare the results by methods, so basically we took graph compounds and did this analysis, UMAP analysis of clustering, we can actually see quite nicely that each method forms its own clusters and that are quite distinct. So what this figure tries to tell you is each method provides you with different classes of compounds, different molecules, and all methods are not overlapping too much, right? So they're quite orthogonal and provide you with valuable insights. If we take a look into the space coverage, which is quite interesting as well, and now we just 3 spaces, explore real space and freedom space, we can also see that each space forms its own clusters. So the overlap of chemical spaces is very small, it's almost negligible, and each space provides you with individual results. So therefore, it makes sense to search in different chemical spaces using your compound of interest. If you now do the same exercise with the libraries, we'll see though that things become more homogeneous, right? So it becomes hard to differentiate. Is this compound from ChemDiff? Is this compound from live chemicals, MQ or MPort? Everything becomes more or less the same. So the results that libraries provide you, as I've mentioned, the decline is quite small. They become indistinctive. So if you have 100 results for one compound from library A and library B, it becomes close to impossible to differentiate which library provided you with those results. So ultimately, the comparison here is chemical spaces provide you with distinct clusters. They are more precise. The structures are more related, not more related, more distinct. They are more focused, more guided. whereas libraries provide you also with stuff that becomes just so dissimilar that it's not possible to distinguish. I mean, in this case, it's not even possible to distinguish if a particular result belongs to this query or this query, because it's just too distinct. For computing times, this whole exercise was quite tedious for us, especially the MQ library. Our algorithms are not designed for enumerated sets. I have to admit this, and everybody should be aware of this, which means that it took us quite some time to compute enumerated sets. We see more than 100 hours for MQ. For the chemical spaces, even given their size, it was close to nothing. It was just like this, right? If you put this into numbers, how many compounds can we screen per minute? You can see here for MPORT, we are reaching the 100 million compounds for all of them. For SpaceLight, which is the fastest one, and 1 million compounds with SpaceMax, and yeah, what is this? I think 70,000 with F-trees. Where in the case of our combinatorial libraries, we start to hit the way larger numbers. I want to count the zeros right now. So ultimately, the takeaway is here: F-trees can be up to 10 million times faster for combinatorial libraries than for enumerated sets. SpaceMax also up to 100 million times faster, and fingerprint searching is 1 million times faster for those investigated. To be fair and transparent here again, those are the numbers of our outdated format. So previously we worked with another file format. The new database format for chemical spaces will be way faster. I would assume approximately 4 to 5 times faster. No, 2 to 3 times faster, approximately.